Hi everyone, today what we're going to do is we're going to take the sequence of A's, T's, C's, and G's in DNA and turn it into the amino acid polypeptide chain. In order to do this accurately, there are 3 steps to follow in this specific order. You're going to transcribe the DNA into an mRNA sequence. In that sequence, you're going to find and circle the start codon, and then following that, you're going to mark each of the triplet codons, and finally use the codon wheel to translate the code into an amino acid sequence. This is a codon wheel. What we see representing our codon is the first letter of the codon here, the second letter of the codon here, the third letter of the codon here, and on the outside are the amino acids. To use the codon wheel, we first need a codon. So we're going to use GGG, and we are going to shift over to the codon wheel. We're going to start right here with the first letter being in the inside of the bullseye. The second letter is in the second layer. It's also a G. The third letter is in the third layer. It's also a G, and it's attached to the amino acid name glycine. Glycine is represented by the codon GGG. You can see that there are 2 other codons that need to be translated, plus 2 questions. So I'm going to ask you to pause the video and answer those questions to the best of your ability. When you're finished, turn the video back on and check your answers. So go ahead and check your answers. I do want to point out to you that UGA is not an amino acid. It is our stop codon, and there are 3 different stop codons. UGA happens to be one of them, but we also have 2 other stop codons, UAA and UAG, and when we find these in our amino acid sequence, we stop. Now we want to follow the rules to be able to do the entire protein synthesis process from transcription all the way to our amino acid. So you would be given a DNA code. Your DNA code is in your notes. It's the same one I have here. And we are going to begin the transcription process. Remember that mRNA is made using DNA as a template. It will fit the bases in according to complementary base pairing rules. So the G on the DNA is going to be matched with a C. The A, if we were making DNA, would be matched with a T, but we're making mRNA, so this is going to be a U. The T matches with an A, and again, the A will be matched with a U. The C will go with a G, and so on. I'm going to ask you to complete this mRNA on your own, so please pause the video. When you are finished, you can start the video up again and then check your answers with the completed mRNA that will be there. This is the completed mRNA. Next, we're going to move on to step B, which is to find and circle the start codon. Now, the start codon on our wheel is not marked, and so we're going to take the time to mark it. There is only one start codon. That start codon is methionine, and it is found in one part of the wheel right here. Methionine has the code AUG. So AUG is always going to be the start codon that we're looking for, and it never changes. So we're going to look for AUG in our mRNA, and we need to check each of the bases in order. So this is not A, but here is A, and we're looking for AUG, which we have found. These 2 bases that are up in the front are ignored, so we're just going to cross those out, but we're going to circle this start codon so that it's very clear where the start of the protein is. Now the next thing that we're going to do is we're going to mark the rest of the codons so that we can read the code. So since the first one is in place, we're simply going to mark that out with a line, and since codons are every 3 bases, we are just going to go right down the length of the mRNA and we're going to mark each of the codons that follow. Now, it is important to note that if you had done C before B, you would have ended up with your first codon being right there. And you can see that that would change every single codon and the entire sequence would be incorrect. So now we use the codon wheel to translate the mRNA into amino acids. We know that our first codon is AUG, and AUG is this amino acid here, which is called methionine. Now that takes up a lot of space, so when we're doing our translating, we use the first 3 letters of the amino acid only. And because many of our sequences will be long, I'm simply going to cross it out to make sure that I know that I've already translated that. My second codon is GCG. So GCG is the amino acid alanine. So I'm going to write ALA. Now, I'm going to ask you to go ahead and continue to translate until you get to the stop codon, in which case you're going to stop. But in the meantime, I'm going to ask you to pause this video, and when you are finished, then you can turn it back on to check that you've translated properly. So if you've done this properly, you should have the sequence met, alla, phi, his, arg, lu. And you'll notice that I have not put the stop in there because stop is not an amino acid. It just means we stop. You'll also notice that there are some extra codons at the end. And like the ones at the beginning, they have come after the stop codon. And so we just ignore them as if they were not there. So this particular amino acid sequence is 7 amino acids long. So hopefully this makes sense. You guys have some practice that you can do past your notes, and you can check your key on Schoology when you are finished.